Okay, during the break, let's go over some of the student questions and earlier 2 weeks or 2 or 3 weeks ago that you had it. Uh, so this first question asking once again about the mechanism by which this covalent bond is formed between two hydrogen and one oxygen. So always this covalent bond is always the electrons. Oxygen had 6 electrons on the outermost electron shell. So in other words, oxygen needs just two more electrons to become fully occupied. Octet rule, 8 electrons in the outermost electron shell. But the arrangement of such oxygen has, so to speak, we have 6 electrons are arranged, so to speak, like this. On the other hand, hydrogen only having 1 electrons, okay? So to become more stable, in the case of hydrogen, it needs one more electron. So hydrogen, okay, let's put it in different color. Oxygen, 6 electrons. Hydrogen, 1 electron. So having this, so both parties are happy in that, like they all have accomplished this the most, like the fulfilled the outermost electron numbers, and this shared electrons are the one actually creating such covalent bond. So we usually designate them by drawing a straight line in between. However, after you having this, one big problem is, unlike their original agreement of like sharing these two electrons equally in between, but actually in reality, most of the electrons are spent over here in the oxygen side because oxygen is more greedy, like so to speak, more electronegative. So if you ask that actual the principle of the covalent bond formation, I have already answered. But if you are like further asking that what happened in the hydrogen bond, and there is some inequality in terms of uh, the charge distribution. So in this area, slightly negative, and this hydrogen side is slightly positive. So this unequal distribution will make the one single water molecule is a polar. And usually water molecule does not exist only alone as a single molecule. In the neighborhood, you also have another such water molecule having the same problem. So between this partially negative and partially positive, you do also have another very subtle attraction between. And this is the principal nature of hydrogen bond. And this very acetyl, so So this is gonna be a partial part of this phospholipid bilayer held together through this hydrophobic. This area is hydrophobic interactions area. And the question is whether this hydrophobic interaction is also held together by van der Waals force, because all other hydrophobic interactions during the lecture, I said that actually the force maintaining such hydrophobic interaction was actually van der Waals force. In the case of this lipid bilayer cell membrane, this structure of a fatty acid tail, this hydrophobic tail of fatty acid, is not really ideal for having such van der Waals interaction. But we cannot say that there is no van der Waals force is there present. Something that's obviously got to be a van der Waals force should be present over there, and that almost certainly should contribute to the stability of the cell membrane, having integrity of this lipid bilayer. However, because of such a unusuality of this is not really conventional van der Waals force maintaining structure. However, still, I'm not saying it is impossible. It should be there some. And you cannot really, it is technically very difficult to concisely measure the existence of such a van der Waals force. But indirectly, what we can infer is that because of that, this membrane structure formed by this lipid bilayer is very flexible because of that. So if this was strictly held by standard van der Waals force, probably this membrane structure, cell membrane structure, should have been much more rigid. That's probably what I can say about. And okay, extra gravy about for this lady, what she had is, it is not a question, but an explained about when I went ahead an enzyme and activation energy. I spent actually a long time about trying to explain the nature of this, the energy of activation in enzymatic action. And then she went on like, okay, the definition of the activation energy is how about enzyme lower the energy required for a reaction to take place. So in other words, she suggests that how about oh my. The definition of the activation energy is the energy required for a reaction to take place. Yeah, obviously, perfectly. No problem at all. However, what I intended during the lecture was to explain in more the factual nature of what it is about the activation energy. It is something like a casual way of explaining what's the activation energy. Of course, it's something that the energy required for a reaction to start, to take place. However, that is not really enough. What is exactly the nature of activation energy? That's what I had to bring bring in the appeal and the hump thing in diagram. In lipids, why the lipids are the most energy dense nutrient? The calorie is when we talk about the energy potential like glucose, every food molecules like lipids and carbohydrate and protein will eventually be used during the digestion procedure and be utilized in the production of, so to speak, this ATP generation. Okay, So in terms of how many calories, this calorie is something slightly different. How many potential energy? How many potential energy is in their particular molecule in terms of a chemical bond? So if we are directly trying to translate this calorie thing in each different macromolecule in their energy potential, sometimes it is not accurate, the conversion. So although certain, and also there are lots of different lipids present. So even if we just, fine, let's say some lipids have a certain amount of a calorie, 9 kilocalorie per gram, okay? For some reason is one of such reason is they have the presence of this long fatty acid will contribute obviously to having more the kilocalorie for the given unit amount of the mass. However, that doesn't necessarily mean that this is the better energy source for carbohydrate. That is something different, conversion and translation. And this is question. So do we have 20 different kinds of protein in our body? No, it is not. What it means is 20 different amino acid. We do have 20 different kind of amino acid participating in assembling different kinds of protein. And speaking of number of different proteins, actually in human body, in human cell, how many different kind of proteins are there? Probably the estimate is about 100,000 different proteins are present in human cells. In other words, we, with only 20 different amino acids and having different combination of such 20 different amino acids, we can create this many different kinds of protein as we see in our real example of our cell. What happens when there is a mismatch in complementary base pairing? Two things. This is perfect match. But what if there is a mismatch? For example, instead of A, we accidentally had something C is there. Okay. And one thing is, then this particular structure doesn't perfectly fit, so that probably this structure will be like stuck out in terms of the dimension. So that is something ugly. Okay, that's immediate consequence. What happens? But that such immediate consequence is not something that really seriously affects us. The DNA structure doesn't really fit very well because of this mismatch, one thing. Then if that happens, some of the patrolling enzyme, surveillance enzyme, will detect this type of mistake and try to correct, okay, to force them into original perfect match. That's something. Another more serious problem is when this area is undetected and then goes through DNA replication. So when this region is being replicated, at least either one is two area, this particular mismatched area will be duplicated and then result newly synthesized area will carry a wrong information in terms of is unlike the original information preserved, but due to this mismatch will create some different wrong, so to speak, wrong DNA information, which now we call this as a, during the DNA replication, this mismatch will generate what? Mutation. So at least half of this replicated DNA will permanently carry this mutated nucleotide. And a lot of times this mutation will probably, they can go unnoticed without any consequence. But if this mutation occur in very critical area of the gene, then the immediate impact can also occur. That's probably can happen potentially. This is not a question, but this is, I like this summary of this particular version of the lecture. This lady summarized very well, so I liked this. Oh, by the way, I forgot. Through this, the attendance scrap paper, as well as some other students through even email while they are presenting their the exam, they have expressed the condolences for my the that the the incidents. So I really deeply thank you, thank for such condolences expressions. So with this, taking this moment, I'd like to thank you, all of such students who also with me in terms of such emotion. Thank you. Okay. And the kimchi, I know kimchi is food with enzyme. That is, I cannot really 100% agree. Perhaps this actually, the kimchi is not the food with enzyme. Kimchi is one kind of a fermented food, fermentation. Fermentation means this particular digestion procedure without oxygen. Under the condition of no oxygen, you try to digest, and then, what do you mean digest? You try to break down your food, okay, the glucose or whatever those food available, and that we call that as a fermentation. And you produce some unusual products through this fermentation, like, for example, alcohol. In the case of yeast, that's why we can enjoy those alcoholic beverages thanks to such particular fermentation behavior of this microorganism like yeast. And some other cases, like some bacteria also ferment and producing different type of a final product. So kimchi, which is being fermented by several specific kinds of yeast, is a fermented food. But some people think this kimchi has food with enzyme, maybe because this, in Korean, this fermentation and the enzyme has very similar vocabulary wise. Fermentation in Korea is called, and enzyme in Korean is this. So this hyo indicate the presence of this enzymatic activities sometime. So maybe that's what this lady has a little bit of confusion about this kimchi is a food with enzyme. No, it is not. And also, some media, especially several years ago, some media has actually misled a lot of people having this fermented food as some food with enzyme. There is no such thing like a food with enzyme. That is totally bogus thing. If you have ribosome only, by the way, the ribosome start with R, it's not L. I do that every time. Then can we do this protein synthesis in the laboratory? The answer is yes, of course. But we do need some more extra. So definitely if we isolate ribosome from some other living source, that is a start. And in the test tube, but ribosome is the core machinery with which we can do this catalysis, the protein building enzymatic activity, because the ribosome itself contains that particular enzyme. However, we do need some other raw material like amino acid, right? So several individual amino acids that we need and some other energy source. As long as we provide all we need into the test tube together with the ribosome in terms of a purified, isolated status, yes, of course. And that is called actually in vitro translation. Translation is protein synthesis procedure. In vitro means in test tube. And this is like actually common practice behavior during the we try to produce a specific desired protein in the laboratory. Okay. So the once again answer is a yes, and it is being done in these days. Okay. Why the DNA and RNA are twisted? Good question. Let's say if this is the line, the core line of DNA or RNA, whatever, and if this is the nitrogenous base inside, and their angle is not 180-degree horizontal, but they are like airplane, the propeller, they are slightly twisted. This is a slightly twisted status. So to fit this slightly twisted, this nitrogen bases, then all this, the axis should turn, okay, twisted naturally to fit them so that they can have a perfect fit hydrogen bond structure. Maybe a similar analogy is if you let your fingernail uncut for like 20 years, what do you think? Then your fingernail will grow straight line? No. I have seen some the odd people like having such a long fingernail, but those all fingernails are all twisted naturally because that is the naturally the most stable structure, and so is the case in the DNA and RNA. This twisted form due to their specific arrangement of the component, and this twisted structure is the most, energetically speaking, most stable structure. That's why. I was so surprised to hear that enzyme is the only thing that speeds up reaction. Yeah, okay. But that's the only thing that enzyme can do. Enzyme is not like some magician, unlike our like, casual perception. Enzyme can only speed up the reaction. but that is really important contribution in actual the success of any given reaction. And also, like to be more accurate, actually enzyme is not the only thing that speeds up the reaction. There are several factors, several other factors that can affect the speed of a reaction. The speed of a reaction we generally designate as the rate of reaction. Reaction rate can be influenced by some other factors, including how many of those substrate molecules you have in a given such location of the reaction, and what's the temperature of this reaction, etc. All can affect overall speed of the enzymatic reaction. Both sucrose and trans fat cannot be broken down by enzyme. That's true, because we don't have any enzyme. Then why is only trans fat hazardous for the health? Why? Why not sucralose? Because sucralose, they advertise that those manufacturers of the sucralose, they advertise that without any harm done, this sucralose can escape our body system. That's what we heard. But trans fat, we obviously heard also too many stories, bad stories about this trans fat. What's the difference? Maybe the difference lies in the lipid versus carbohydrate. Carbohydrate is hydrophilic, right? So it is very difficult for them to enter the inside of our cell. So usually majority of them, if they cannot be found, I mean, if the proper enzyme cannot be found to break them down, and what we can expect is they usually pass through our body system untouched. But the lipid is different. Lipid can easily pass through our cell membrane, and they can remain inside of our body, especially in our bloodstream. So maybe that is the reason why this trans fat has more serious adverse effects. And besides, because of that, actually, some environmental activists claim that the sucralose is not something that harmless, but sucralose can potentially have a serious damage on our liver system, which means this sucralose can go into our liver to have any opportunity to be broken down or any similar thing. But we are not really sure how accurate those reports are, but at least some of the concerns are there. But obviously, of course, even if those concerns are there and some significant portion are true, but still the adverse effect, the magnitude of this bad effect done by trans fat is much larger. That's it? Oh, okay. So we can now get into human evolution, the origin. So by now we know that chimpanzee is our closest relatives, which means we share the common ancestor with chimpanzee in the most recent history. Fine. So what? Let's go. So with chimpanzee and human, there are certain specific groups that are all very similar. This monkey-like, chimpanzee-like group of animals together that share at least relatively recent history that share the common ancestors. And then we call them as a primates. Okay? Several different primates photographs. Fine. So if we draw the phylogenetic chart based upon its, these are some relative relatedness of humans and chimpanzees, like some other primates. But what about, okay, here, so we all know that chimpanzee over here and us humans are the two existing closest relatives altogether. But on the other hand, we also know that even chimpanzee and human, we do not even share the common genus. So they are very, so to speak, distantly, remotely related than us. And here, what about this? Something like Australopithecus, which no longer exists. This. So what about this? Do we have like much more closely related relatives actually have existed before? Or then what's the relationship uh, between us and some of the extinct, some human-like, the should they call it a primate or something else? Is probably the opening question of this particular chapter. So, speaking of this common ancestor, so we split from chimpanzee, okay? Let's say, okay, we didn't have any other further, more closely related, the common ancestor, let's assume. Even with that, the split point, so estimated like as 4 to 6 million years ago. And then in between, after that, we had the evidence, the fossil record evidence have shown that some other, like our ancestral forms of such human-like beings, at least several of very renowned, famous things, including the Australopithecus, Homo habilis, Homo erectus, Neanderthals, and Homo sapiens. Traditionally, all these different names of ancient humans were arranged in, so to speak, chronological order. Like, so Australopithecus in the oldest, and the Homo habilis is something that a little more developed version of Australia, so like evolved one. And with that, Homo erectus, and then finally into Neanderthals and Homo sapiens, like in this more and more recent form of human. Partially that's true, but also partially that is not entirely 100% absolutely accurate one. But anyway, Homo habilis means this habilis they suggest, imply that the tools. So this, at the stage of this Homo habilis, these people, or should I call them as a people? Whatever. Or this, but they're not animals, so I will call them as a people. So this Homo habilis are the first one who began to use these tools. And what about this Homo erectus? Erectus means they stand upright for the first time. So even Homo habilis was like crawling like this, but still they were able to utilize the hand tools. And Neanderthals are, they are so famous, and you know what they are. Okay, so this, the more and more developed form, brain size become larger and larger, and this is also a very stereotyped, not 100% accurate one. So upright, being able to stand upright and walk with these two legs, we call this a bipedalism, bipedal. And the larger and larger brain size, which is measured by cranial size, are the two kind of like two measuring stick, so to speak, of how advanced in terms of evolution, how advanced a specific type of a human, ancient humans being. And obviously, the reason there is a very good reason why these two particular characters were very important. So anyway, all of those, whatever, the Australopithecus, Homo habilis, Erectus, whatever, they were all actually, where did they come from in terms of their origin? It turned out that they were all originated from Africa. Surprising. So, like before their migration out of Africa, that means the rest of the land on Earth, there were no human ever lived. Probably that's what we call the Africa. Africa is our holy origin, holy place. So it's kind of we take this as a fact. All living, present living humans are all came from originally from Africa. And who are the one who actually began the migratory activity at the stage of this Homo erectus? Some group, not all, but some group of Homo erectus once originally living in Africa, they contemplated the moving out. Should we move out of this Africa? This is too boring. Let's go out of this Africa. And they did. And so they went into Europe and Asia. Like that it turned out to be about 1 million years ago. But unfortunately, they did not really do very well. So virtually almost all of them are extinct, okay, except these Neanderthals are the sole survivors, okay. Obviously, of course, between Homo erectus and Neanderthals, there might have some more branches of newly differentiated human. I'm not saying there were only two different kinds of directly all these Homo erectus were able to evolve into the Neanderthals. That shouldn't be the case. But if we follow only the mainstream, that is the case, is what I am saying. So anyway, but then another such brave migratory attempt also had been made in more recent past, like 150,000 to about 200,000 years ago, this time, once again in Africa. So this time, this small group of the human migrated out of Africa this time was the actually direct ancestor of us. So that's what this chart tried to indicate. It's out of Africa theory. There are two different versions of this origin, but the previous one was incorrect one, so I just entirely skipped. Okay, so this out of Africa theory, it was turned out to be the correct one. So there are here it states that there are two independent events of migration from Africa. That's the point. Once earlier in Homo erectus, okay, and all extinct, but some legacy remain in the form of Neanderthals. But much later, this Homo sapiens, now us, our ancestor, also did, and this time they have succeeded greatly. So they spread all over the area of entire Earth, is what this out of Africa theory. But the one thing is, then if that was the case, when they did this, okay, at least in Europe, at least, and some part of Asia, the pre-existing inhabitant must have been there. Who? Neanderthals. Neanderthals were living there. And then newcomers from Africa, they just showed up and hello, hey, we are new migrant from Africa, and let's shake hands and live together. Actually, I don't know what happened. If they fight, if they fought each other and they just live happily for a long time, I don't know actually. But anyway, coexist for a long time. For a significant period of time, they have lived together on the region. Okay? That's the point. So this is the actual what happened when this migration occurred. Eventually, they all spread into to all over the place of this current present land is what this map shows. And this, not only that, some of the other, so here, Australopithecus and some other miscellaneous ancient humans were present over here, and in Homo habilis and some other humans that I did not mention, so which indicate that there are some other miscellaneous. Always this technical thing happens. I had to pull out this camera thing because without. Is that the camera thing? Camera is working still? Oh, okay, still camera is working. So the only thing is not working is the audio. Okay, I see. I'm not sure whether this recording will just resume or not, but let's keep the fingers crossed. It's not it's not your problem, so who cares? Okay, so anyway, and this. Recent, for example, let's just try to focus on this more recent Homo species, including Homo sapiens and Homo floresiensis. The nickname for this Homo floresiensis is a hobbit. This hobbit, very small people, okay? And Homo neanderthalensis, who are they? They are Neanderthals. But some other times they are called, instead of Neanderthals, they are called Homo neanderthalensis. Okay? Or some other times, this name for Homo neanderthalensis, instead of, they are called Homo sapiens neanderthalensis. Surprising? I will explain in a minute. And Denisovans and Heidelberg, these are at least, there must be some other recent Homo, or so to speak, species. Should I call species? I'm not really sure, but like early humans, human-like people, they were present. But anyway, the point is they all coexisted together during the overlapping period of modern times. So what happened to them? The point is, so how do we? So once notice that these are the one at least in the form of the fossil record. Okay, so now we know that these different groups of humans, ancient humans, existed. Then how do we know? Okay. They are related. Yes. Okay. This Homo, so to speak, this hobbit, are they close to us in terms of origin or evolutionary origin? Or what about these Neanderthals? Which ones between Neanderthals and Denisovans or the hobbits? Which one is the most closely related to us? Okay. How can we determine such thing, such relative relatedness? Just by looking at and comparing the shape, relative shape, morphology, so to speak, of these fossils. Is that it?
DNA?
DNA, yes, exactly. DNA has been very instrumental tool in determining such relative relatedness. And another thing is, not only that, we are all the descendant of this particular version of the human, Homo sapiens. okay, everybody. But we all know that we all different, okay, in these days. So what's the relative relatedness, so to speak, like as a Korean, okay? Maybe you might be interested in knowing that. Are we more closely related with the Japanese or like Mongolians or Chinese? Well, which one? And how do we know such thing? Just by hunch? We should be very much tightly related with the Mongolians. But is there any proof? That is this, actually provided by DNA. In particular, among different DNA, mitochondrial DNA and Y chromosome DNA have been very useful. Why these particular two different kind of DNA? Let's see. So in this occasion, we are trying to compare the actual DNA sequence, okay? But not entire DNA sequence of a human cell, which is like 46 or 23 different chromosomes, entire DNA sequence. If we align them in linear fashion, it will be something like 3 billion nucleotide base pair. Are we going to read all 3 billion and then compare? In some occasions, we do that, if that's necessary. But in this occasion, we don't even have to do that thing. Then what's the alternative? By the way, without audio, it will be very, very difficult. How am I supposed to solve this problem? Okay. So this sequence comparison has been used in determining the origin of modern humans, as I went over, and also interrelationship among today's human races. And for that, one particular useful component has been mitochondrial DNA. If you were surprised that mitochondria even contains DNA, yes, they do. Mitochondria has their own independent DNA, right? Mitochondria is actually the organelle. And what business brought their like ability of having their own DNA is very surprising. That is something related with the so-called this endosymbiosis theory of this origin of mitochondria and chloroplast as their origin, because they were a long time ago our the theory of this endosymbiosis theory is that they were independently living bacteria, and then they were somehow captured by a big eukaryote and then became co-inhabiting them, living together. That's what this endosymbiosis theory. In next chapter, we are going to take a look at in more detail about such endosymbiosis theory. But for now, let's just focus on and then try to accept the fact that mitochondria, even they are organelle, they do have their own independent genome, gene, DNA. Okay. So like mitochondria are shaped like this, and in a given animal and plant cell, they have multiple copies of some cells. Some animal or some plant cell have a single cell contain about 100 or several hundred multiples copies of mitochondria, and some other cell have a few of them, fewer than 100. But anyway, each mitochondrion, should I say, because it is a singular, mitochondrion, even contains more than one copy of DNA, but several multiple copies of their DNA. They are all identical duplicates, copies of their DNA. And their DNA is unique in that they have this circular shape, unlike our DNA, which is linear. their DNA is all in circle form. And this is actually characteristic of all prokaryotic cells' DNA. All bacterial cells' DNA is this circular form. And mitochondrial DNA also has this circular form. And anyway, so they have their own, obviously, they have their own DNA. And we are comparing each DNA sequence of each individual cell's DNA with some other individuals' mitochondrial DNA and how similar they are in terms of their DNA sequence. Okay. And the advantage of using this mitochondria as a DNA sequence comparison is, one is this. They don't have any recombination. What do you mean by recombination? This is recombination. Okay, in the blue chromosome and red chromosome, each represent both maternal and paternal. So these two chromosomes must be what? I went over this particular vocabulary in the first session, which is start with H, homologous. So these are homologous chromosomes, right? And during meiosis, they do have this volume dense. And someone mentioned that it's duplicating. Obviously, of course, just with the onset of DNA replication, every chromosome has its doubling up duplication form. So temporarily they form this X-shaped chromosome. But what's important is during this volume dense, so in the event of meeting this homologous chromosome pair and line up, they have this crossing over of their legs and arms. This is what we call crossing over. And the result is swapping some of the region of the chromosome region between the two homologous chromosomes. The consequence is, even though these two homologous chromosomes are homologous, so basically same identical chromosome, however, if you think of the actual origin, this homologous chromosome came from father. The fathers gene have been for a long time have been living independently of that of mother's gene. So even if these two chromosomes are same kind of chromosome, do you expect that exact DNA sequence of this area would be identical or something somewhat different? Somewhat different, obviously, because these two, mother and father, have been lived for a long time, totally unrelated. So during which time, a lot of independent mutation must have occurred, which created some DNA sequence variation, right? That's exactly what has happened. Okay. So this swapping will generate what? More variation. That's what we call this recombination. And this recombination only occurs during meiosis. During mitosis, it cannot happen because these homologous chromosomes do not meet at all, ever. So they don't have any opportunity of this, hey, let's exchange this thing. Does not happen. Only during meiosis, which is the production of gamete, only occur this recombination. So, in other words, if we rely on the DNA sequence comparison of chromosomal copy of the DNA, then inadvertently we invite some proportion of this recombination. And the rationale of DNA sequence comparison is what? All these differences in DNA sequence comparison came from what? Mutation. It's the absolute premise. And only then we can absolutely accurately determine the relative, in terms of relative relatedness in terms of evolution. Because this mutation will occur in terms of function over time. But this recombination will have an impact, a confounding effect, like clouding the accuracy of such thing. Although it is negligible, obviously. This recombination is overall scheme, it is negligible. However, still, it's there. So recombination will contribute some inaccuracies of this estimation of evolutionary relatedness, relying on DNA sequence comparison. However, mitochondrial DNA do not have such a thing, because mitochondrial DNA is basically haploid. They don't have any homologous thing. They don't have any recombination event. That's what it means. So only this nuclear DNA is undergo recombination during meiosis, but mitochondrial DNA do not. Okay. So what's the advantage of no recombination? So in mitochondrial DNA comparison, all those sequence differences, the sequence variation, is coming from mutation only. So more accurate. So any sequence differences more directly, okay, translated into actual differences in the evolution. Why? Because they have no homologous chromosome. They are basically haploid. Like their origin is prokaryotic. Bacterial DNA, they don't have any homologous chromosome. Only eukaryotes engaging in the sexual reproduction have this. Okay. And another very important advantage of using mitochondrial DNA is it has many copies. A single cell contains many mitochondria, even several hundreds. Okay. Even each of several hundred mitochondria contains also multiple copies of the identical DNAs. So let's say you discovered some fossil from very, very old, ancient. So it must be something that belonged to Neanderthals or like Homo or Heidelbergs or whatever, some other form of extinct humans. And then you try to extract some DNA. Fossils are basically like bones. You try to extract some DNA from bone. How many intact DNA can you recover? So especially on such an occasion, this trying to use mitochondrial DNA instead of chromosomal DNA should be huge advantage, right? So will obviously definitely facilitate the speed of this analysis due to this. Another thing is this mitochondrial DNA, actually, mitochondria are only in most of organisms, especially in animals, all these mitochondria are coming from mother side. Father does not. Father does not contribute at all any mitochondria. Even what's happening is, in the sperm, mitochondria is the one actually generating the energy, ATP. So sperm must contain enough copies of mitochondria, obviously. When sperm fuse and then enter the egg, they also bring in their own version of mitochondria. And you know what? The fate of this paternal father side mitochondria, they are all get killed directly. Once the fertilization succeeds, then each of the remaining present alive these paternal mitochondria are deliberately killed so that all those remaining mitochondria must be maternal origin. So in this way, we can only follow the lineage of the female side of these evolutionary traces. So sometimes we call such kind of endeavor as a project as a mitochondrial Eve project, so that by comparing all those different female mitochondrial DNA, we can trace down back to or only the female origin. And actually, it turned out that the origin of all these remaining existing humans, the females, were all originate from very only handful, very few number of original female from Africa. It turned out that. So not many people actually have migrated out of Africa, but originally the number of individuals who crossed the Africa between, and Europe channel was very small number of people. Okay, turned out that. And on similarly, another very useful DNA resource for such evolutionary DNA sequence comparison is the human Y chromosome. Y chromosome also possesses the characteristics of a no recombination. Even though my Y chromosome is in the nucleus, but due to its very unique structure, it only belongs to the case of course, a human, although. In frogs, for example, X and Y chromosomes are like truly, virtually homologous. They look alike, and the genes present between Y and X are kinds of genes are identical. So that's the definition of a homologous. But in the case of human, Y and X chromosome, you decide. This pathetic Y chromosome is very reduced shape, size, compared with X chromosome. And even the kinds of gene present between Y and X are entirely, totally different. So they are not really truly homologous. They don't look like, and the kind of gene presence are not at all the same, but simply the only kind of trace that still they can behave as homologous is even with that, they line up together during meiosis. That's all. But anyway, even if they line up, will it be possible for them to have the crossing over, recombination? No. There is no similar region, so they can't do this. So that's why Y chromosome cannot participate any recombination. But if you happen to be a female, you can freely have such recombination because you have 2 copies of X chromosome. So recombination can happen in female. But in male, no way. You can't have any recombination. So some scientists actually predicted, because based upon this, eventually all Y chromosome will vanish in the human population. Yes? Okay, maybe. So then there won't be any remaining male. No Y chromosome, no male. Then until then, yeah, you have to struggle. Okay. But still, there are some ugly males still present, so you have to put up with them. Okay. So anyway, so this no recombination is actually ideal property to become to be used as the sequence comparison source for this evolutionary determination. So, like, similarly to the mitochondria Eve project, sometimes we call this as a Y chromosome Adam project. So like Adam and Eve in the scripture Bible, the first man and woman. So that's the reason why Y and mitochondrial DNA have been useful in determination. So that's that. So many valuable knowledge have been obtained through the analysis of a sequence comparison of Y chromosome as well as mitochondrial DNA. And this is totally different story about Neanderthal Genome Project. It's been started with the Germans, the renowned Max Planck Institute, and within very short, remarkably, impressively short period of this time, they have finished this project, whatever. And the bottom line is they finished in reading entire DNA sequence of Neanderthals. If you imagine, there are no present living Neanderthal individual, not a single Neanderthal individual exists, but still they were able to accomplish. That means all those available fossils, they were able to extract some pieces, small little bits of pieces of DNA traces, and then they were able to combine, put together, and then reassemble entire whole intact DNA, which is a 3 billion, just like us. Neanderthal also had 3 billion DNA bases, like exactly like Homo sapiens, and then they finished reading. Once they accomplished, then now they can compare one by one, compare entire whole map between Homo sapiens, human, we, and Neanderthals. Okay. And then it turned out that, wow, 99%, 7% identity in terms of DNA sequence. 99.7% identical, which means almost 100% identical between human and Neanderthals. How can we find any differences? And in reference with that, between chimpanzee and human, the similarity is 98.8%. That is also very, very significant. But still, we know that there are too much difference exists between chimpanzee and human with such a small differences. So with this 0.3% difference still indicate the big enough difference between human, the Homo sapiens, and the Neanderthal, obviously. But more important thing is this interbreeding. Once you finish this DNA reading, now you know that, oops, our DNA, some portion of our DNA actually originally came from them. So what do you mean? How do we end up having their DNA in us? What else? Interbred. So crossbreeding. They made it together, which means then how can we put this us Homo sapiens and Neanderthals as two separate species? We can't. We should put these two as a single species, same species, because they were able to interbreed. That's why some scientists actually designate the scientific name of us as a Homo sapiens sapiens, whereas Neanderthals, we'll call them Homo sapiens neanderthalensis. So we are both Neanderthals and humans are the same species, but the only difference is like the third names, the subspecies names are the difference. Kind of trying to indicate that. So gene exchange through interbreeding, not only with Neanderthals, but it turned out that prevalent at the time among these different humans with Neanderthals or with Denisovans and these hobbits and Heidelbergiensis, whatever they are, all these ancient humans with us, they have some gene exchange have occurred. But obviously, the area and the timing of coexistence been greatest with Neanderthals, so the legacy from Neanderthals must be greater in our gene, and that is the case. Okay? So these are some relative heat map of the relative distribution of these genes, like how much of these Neanderthal genes in terms of these different populations still contains, relatively speaking, etc. But one thing, important thing is here in this Africa region, these are still the fewest. These Africans, original native Africans, had the least amount of this gene exchange. That is a proof of, because these are those who did not live out of this Africa, the descendant. So there, actually the genetic diversity of these Africans are the greatest. Whereas the amount of this gene exchange between other humans are the least in Africans. Why the genetic diversity are the greatest in Africans? That means only few, very small number of people have migrated out of Africa. And all the descendants widespread in these are the direct descendants, offspring of this originally very small population. That's why the genetic diversity of the outside region of Africa is the smaller than the inside mainland Africa, where still the majority of them remain unchanged, and they have much diversified gene exchanges. That's why they have greater genetic diversity. That's a good explanation. Now, the final thing, final message about this, the interbreeding is that, so like I said, a lot of traces we have that evidence that the origin, some of these genes must have originated from Neanderthals, including, very interestingly, including like genes causing depression, genes responsible for allergy, genes responsible for the heart attack, the circulatory problem, genes responsible for causing diabetes and addiction, etc. All those nasty, bad genes are actually originated from Neanderthals. Wow, shoot. So if you happen to be something later, suffer from diabetes or some, I cannot really put up with this nicotine addiction or anything, it's not your fault. It's not your fault, it's Neanderthals' fault. Yeah, right. So you can feel less guilt. Okay, great. What's another message, take a message of this? So all these bad genes, actually, Neanderthals had all these bad genes. How could they survive with such a bad genes? So no wonder why they all extinct. Okay, so okay, the justice served. Is that the correct interpretation of this, or something else? Is there any other way we can predict this? Is that my final question? Final take-home message of this lecture. What do you think? So those bad genes are the one actually that belong to Neanderthals. So they are the inferior. Neanderthals are the inferior, more primitive version of human, which is indicated by this. They contain such a bad genes. So that's why we outcompeted. So they are all eliminated. Is that so? Or any other version of interpretation is possible? Oh, no, not yet. I always thank you, but not yet. The environment. You can't really think how ignorant if you think that, oh, those Neanderthals are such an inferior, so they carried inferior genes. No. If that was the case, Neanderthals must have been eliminated. But the reason why they were able to maintain their uh, roots and their legacy for such a long time on there. Okay, let's say some of the genes that we carry, okay, that gave us a lot of the troubles, okay, or originally came from Neanderthals, doesn't mean that at that time when they lived there also had the exact same effect. No way. It's always the environment. For example, allergy. We have a lot of us has a trouble with this allergy problem. What is allergy basically? It's a highly overreactive immune response, highly sensitive immune response. But if Neanderthal had the same type of such property of the genes which causes the allergy, would it have been such a problematic at the time? Not necessarily, because highly sensitive, overreacting immune response must have been actually beneficial. Because there, at a long, long time ago, when they lived, at the time, they didn't have any medicine, no antibiotics, etc. So if they had a stronger, more sensitive, quicker immune response, it would have been more beneficial, advantageous. But with that gene, when they were put in this present day's scenario condition, then it could cause some unexpected problem. Addiction is too, and depression is also. Their environment, basically whole open outside environment, would be totally different with this modern days, basically captive environment. So kind of such property must have actually triggered some undesirable effect of like giving some the depression like stuff. So many the examples of explanation are not only this depression analogy but all these the basically responsible genes we can interpret different ways so that all these genes actually must have played positively to them. We can all explain, and I have some list of explanation, but I did not bring up here today because of this time frame. If some of you are interested in, I can actually send you some original copy of this thing. So, bottom line is, it's environment, not the gene itself, that has a different occasion of the responses or consequences. It probably should be correct interpretation and also take home message of today's lecture. Okay, so with that, have a nice weekend.